Every year, thousands of dogs have their chests cracked open, their arteries tied off, and pacemakers implanted to force these dogs' heart to beat abnormally fast, all to create heart failure in a lab. The same dogs that curl up at the foot of your bed. And here's the thing, that artificially induced disease isn't the same as the one that develops in humans over years. So what are we actually learning? And at what cost? Dr. Charu Chandrasekera spent 14 years doing animal research before founding Canada's first center dedicated to replacing it with human-based science. This is Puppies, Pandemics, and Public Health. Let's join the conversation between Dr. Lieberman and Dr. Chandrasekera about why the experiments we're told are necessary may not be necessary at all.
Welcome to another episode of Puppies, Pandemics, and Public Health. I am Dr. Johnny Lieberman, and I'm pleased to welcome onto the show today, Charu. If there were one thing that you want to make sure that our audience does not miss from everything we're going to cover today, what would it be?
That's not an easy question to answer. There are a lot of things I want our audience to know, but I think the big message is that every animal experiment that is labeled, peer-reviewed, approved, and defended as necessary is not, in fact, necessary. I believe that animal experiments are glorified, and there's no need for that anymore. They still have a role to play in our society until we end the use of animals altogether. But until then, every single experiment that is labeled and defended and approved as necessary is not actually necessary.
That's a discussion and a claim, which I think most people, I think that's brand new information to them. I think a lot of our listening audience, a lot of people in general, feel like animal research, experimenting on animals, which is largely what we'll be discussing today, is crucial. I think the understanding is that it's the research done is crucial for medical progress. The experiments being done on a day-to-day basis are highly valid and give us so much good information. But I think that's not by any means the whole story or even an accurate story. And Charu, you've experimented on animals before, correct?
Yes, I have. And to go back to your first point, just for a minute, animals have made monumental contributions to the life sciences, and we can all agree on that. The discovery of insulin, or even the fundamental knowledge that we see in our textbooks today, that knowledge has been derived through a lot of animal experimentation, but it is also this extensive animal experimentation that taught us that this knowledge isn't complete. This knowledge has colossal limitations. That's the part that we really have to understand. Yes, they have contributed. We should never ignore that fact. But at the same time, we have to acknowledge the limitations, because this is not 1921 when we had to use some dogs to discover insulin. We are in a different century, different era, different technologies at our disposal. So I just had to say that when you brought that up. But going to my story, yes, I started out as an animal researcher. Right after my PhD, I had to make this decision, and I really wanted to become a well-rounded scientist. So I intentionally went into a cardiovascular research laboratory for my postdoctoral training, actually 2 different laboratories, during my postdoctoral training to become a well-rounded scientist by learning how to do animal research. And it was during this time that I experienced firsthand these colossal limitations and the problems with this culture of animal testing.
And so, Charu, I'm going to interrupt here just for our audience. Share, what was your training before that?
My PhD is in biochemistry and molecular biology. So I wanted to go into cardiovascular research because the heart is my favorite organ. And it was one of those things I thought I would go in and get that postdoctoral training after my PhD and then have my own heart failure research lab doing animal experiments. And that's, I imagined like a normal academic research career or even transition to industry or whatever at that time. But the primary thing was that I thought that animal experimentation was necessary to become a well-rounded scientist, because that's what's ingrained in you in grad school, in this culture of science.
Not just to become a good scientist, I think, but to make a significant contribution and to help medical science, right? Is that also part of it? That's part of that norm in the culture?
Absolutely, right? That's what we are taught. That is still the primary message. Animal models are the gold standard. You need to work with them. And oftentimes in this culture of science, we are asked to produce data, right? We have a scientific culture ingrained in animal research, a culture that requires you to validate your human data using animal models, a culture obsessed with curing disease in animals. So that's where we are. I mean, this was 20 years ago now, right? So it was normal for me at that time. So I went into cardiovascular research. I worked with primarily mouse and rat models of cardiovascular disease. When you're in a research environment, you don't just work in your own lab, you get exposed to everything around you. So I had seen dog models of heart disease, rabbit models, and many other types of animal disease
So,
models.
Charu, I'm going to ask you to pause here again
Yeah.
to paint a little bit more of a picture for the audience. So when you're saying, for example, a dog model of cardiovascular disease, what's actually going on? What are they doing to the animals? What do they hope to gain from it? What knowledge? And then how applicable has that been?
So most of the time, the research that people do with animals can be divided into two main categories. One of them is acquiring fundamental knowledge about biological processes, what we call basic biomedical research. And the other one is safety testing, right, for drug development, for chemical safety, and other things. So those are two different categories of animal use. And the one that I'm referring to right now that I witnessed firsthand is the former, using animal models to understand disease mechanisms, to understand physiological processes in health and disease. And so the way that this is done is, so if you want to create a heart failure dog model or a rat model, you have to first induce this disease in these animals. And if you want Animal Research 101, there are four primary ways of inducing disease in animals. You can surgically remove an organ. So if you want to create diabetes, for example, you can, surgically remove the pancreas of a rat and it will immediately go into a diabetic state. I mean, within a couple of days, you pretty much get that. And then you can chemically destroy an organ. And then you could genetically modify these animals and also do dietary manipulation. So if you want to create an obesogenic model, create these obese animals, you can feed them a high-fat diet. And you can combine all of these different methods in many different ways to create the model that you want. So in particular for heart failure, a couple of the most common ones are where you open the chest and you isolate one of the main arteries in the heart and you put a string, essentially like a surgical string around it, a suture, and you tighten it so that you restrict the blood flow to the heart. You occlude an artery, so you restrict the blood flow, or you can install or surgically insert a pacemaker and crank it up to an abnormal beat. For dogs, twice their heartbeat, and you can let it pace. It's called pacing-induced heart failure. And you can let it go for weeks on end, and the animals do go into heart failure within a certain period of time. So there are different ways to induce these kinds of diseases. And the idea is that once the animal reaches that state, you can do different things. You can try to understand the molecular basis. So, take the heart out, look at like the DNA, the proteins, the biochemical pathways and other things, or you can infuse it with a drug. There are lots of different things that you can do afterward, but that's how these animal models are created.
We'll talk about heart failure for a second. So perhaps people, friends, family who've had heart failure. That develops typically over years, if not decades of time. Sometimes it develops acutely after an acute event, such as a heart attack or myocarditis or some other event. But heart failure develops when you have a heart which normally pumps, right? It takes in blood, it pumps blood to the rest of the body. It's got a certain rate it's supposed to go, a certain amount it's supposed to contract. Heart failure develops for a variety of reasons, and the heart, instead of pumping like this at its normal rate, maybe just goes like this, right? It doesn't pump the way it's supposed to kind of try and simplify it. And that comes with a lot of complications. These patients who have heart failure, especially when it's bad, they suffer with fluid overload. They get fluid in their legs. They have trouble breathing, trouble exercising. It's a long list of problems, and that's one of the reasons why it's a target for trying to study and develop better treatments for, ways to prevent it. And so I bring that up because just listening to Charu share this story about what happens, what she described is it happens to dogs. The dogs are the same kind of pet that we saw just back here in the back of my screen. The same dog you have in your homes, they're being induced. They're given heart failure by these mechanisms. That's painful. It's suffering constantly to get to this state. And as an MD, and I think this is something that I think a lot of MDs don't even know, including myself until recently, to study a disease that's created artificially in whether it's a human or an animal, it's not the same disease process. If you put a pacemaker and make it go at 200 beats, the heart's going to tire out, just like humans who have heart rates that go way too fast. You get heart failure from that. And so this model, the ones that Charu described, they're horrific sounding, and they're done by the thousands day by day to create these models which are artificial. And then, as I think Charu might go on and explain, the results that you obtain from that may or may not even be of any clinical benefit to humans.
Yeah, so I mean, you brought up a number of different points. So one of the things is that we are not really recreating the human disease. We are capturing a snapshot of it. That's the biggest problem, right? Because these are complex multifactorial diseases that are influenced by our genetics, our ethnicity, our, lifestyle. There's so many different factors that affect the development of diseases like heart failure that develop over a long period of time, that it varies depending on your sex, your ethnicity, all of that stuff. Like me, as a woman of South Asian origin, like I'm at the highest risk level for diabetes and diabetic cardiomyopathy and things like that. And so it differs. So which mouse is going to represent my identity, my biology, right? So you have to think about how well these animal models are able to capture our biology. Let me tell you, a white mouse isn't going to capture white people biology, and a brown mouse isn't going to catch mine. It doesn't work like that. And so, yes, so there are technological limitations to these, diseases that we create overnight. They do not resemble the human disease, only a snapshot of it. And there are other biological variability like age and sex. And, most of these animals are used at a certain age. So 10 weeks, like most of the work that we did, the mice were 10 weeks old. And if you were to use animals that were 8 weeks or 12 weeks, you would see significant differences in the results. And this is one of the common things, that when you close down your lab for 2 to 3 weeks over Christmas break, most animals are sacrificed by the dozens because you don't want them to get older during the Christmas break, because when you come back in January, their biology is not going to be the same, that you have to keep all of your parameters the same. So there are, I could talk about this for like 5 hours, but there are so many different things to think about. And then at the end of the day, what does it all come down to? Despite decades of extensive research using these animal models, be it heart failure, Alzheimer's, spinal cord injury, ALS, I don't know, pretty much every single disease prevalent in our society today, you know what the ultimate result is? It's over 95% of drugs tested to be safe and effective in animals fail in human clinical trials. For Alzheimer's, it's 99.6% failure rate. Every single major clinical trial over the last decade has failed. And the same thing, what do we have as effective disease-modifying treatments for heart failure? That's the thing. You have a handful of things that can do something to improve your quality of life, but most of these therapeutics that we have today don't really they don't reverse the disease, they don't stop the progression. Yes, it can keep you comfortable, it can maybe slow down a little bit, it depends on the disease and the type of drugs we have available. But at the end of the day, is this what we're calling progress? Is this enough? That's the question I would like to ask people. Are you happy with it? And then, so that's one thing. So things that are done in animals don't necessarily translate to humans. What about the other way around? Are we missing out on certain things because we have now, but what if we're missing out on these drugs that could potentially be very beneficial to humans, but it wouldn't even go through the pipeline because we are seeing some unnecessary side effects in these animals? I mean, the best example I'm sure that you've brought up before is also aspirin. If it were to go through the regulatory process today to get approved, it would never be approved because aspirin causes birth defects in many laboratory animal species, but it's safe for consumption during every stage of human pregnancy. So there are lots of examples of these different things where it's something that works in animals, failed in humans, and vice versa.
You're taking me back. I'm going to share a little bit of my own history of deciding my medical career pathway, which I hadn't thought of in a while, about how so many of the medications that we have, which do get approved, are really for disease management. They're not curing diseases. And I mention that because I realized that 20 plus years ago, when I was an internal medicine resident trying to figure out if I wanted to specialize or if I wanted to do general internal medicine or even non-clinical work. But I realized 20 plus years ago, and I'm mentioning it because so little has changed with animal research, that 20 years ago, it was disease pulmonology, for example. The bulk of their practice is what? It's emphysema. It's COPD. It's a disease which is not reversible. After years of smoking or being exposed to whatever chemicals you breathe in, or sometimes it's genetic, the lung tissue disintegrates, the airways don't work and exchange gas like they used to, and there's no way to reverse it. And I found that extremely frustrating, that that's a disease management process, and that hasn't changed in 30 years, despite all the animal research being done. Same thing for heart failure. It's almost the same. Yes, we have new medications, many of them on the market, but nothing to reverse heart failure, nothing to cure it. And that's going back to when I was an internal medicine resident. And as a resident, I found that extremely frustrating, and something my personal choice, didn't want to spend 30 plus years in a career just managing symptoms. I found that frustrating, and looking back at it now, thanks to Charu bringing that up, 30 years roughly of animal research, tens or if not hundreds of billions of dollars invested into it, decades of researcher valuable time in labs studying disease states, and not to mention all the animals sacrificed. For what? At what cost is all this being done? It's symptom management. And so every specialty, or most, have a disease that there's no reversal for. And that hasn't changed in at least 20 years, if not 30, maybe longer. So I wanted to bring that up, to that really emphasizes and underscores the point which Charu brought up about so much of so little actually gets to humans, and then very little, if any of it, can actually reverse disease. So what is the value of sacrificing all these animals, wasting all this money and valuable time on medications which don't do very much?
That's a question we all need to sit down and ask ourselves, right? We can't do a certain type of research just for the sake of it. If we are going to do animal research, there are so many different factors that need to be taken into consideration. And this is the thing, are we going to just simply do any kind of research? And this goes back to my main point, right? So every experiment that's labeled and approved and defended as necessary is not necessary, and they should not be happening.
Why is it, Charu? Why are so many researchers, you've been on the inside, I haven't. Why is this? It's like, okay, we'll do another, have another 50 rats or whatever. We're going to do this study, and in their mind, they're thinking we're going to come up with worthless results, maybe, but we're going to do it anyway. Why? I feel like that's the culture, to stick with what we've got. Can you shed any light? Maybe I'm way off base, but why is it like that?
It's the way we've always done it. And that it's the culture. It is like there's no other logical explanation other than it's the culture that we have embraced for the past century. A culture, like I said, ingrained in animal research, a culture obsessed with curing disease in animals, a culture that disregards human data as anecdotal. It's crazy, and this is one of the things, and I think you've read about this in recent literature, real phenomenon called animal methods bias, where when you submit a grant or a paper for publication, your peers, other scientists who are, very intelligent and accomplished, all that, look at that and they would dismiss. And sometimes if you have human data, that contradict animal data, you're oftentimes asked to go back and repeat your human data.
Yeah, that makes no sense. No sense.
It's real. And there's like, what are we truly defending? Can you think of any other industry on this planet that would accept a 90% failure rate? And the biggest issue for me as a scientist, a Western trained scientist, is that, like, how much longer are we going to ignore that evidence? And if the evidence, like, science is an evidence-based enterprise, not a belief system. The evidence is before you. So why aren't we dealing with it in a more systematic, pragmatic way? Animals should not be the first resort. That if you have, let's say you're an academic, you want to study Alzheimer's disease or whatever, disease X, and you come up with this beautiful hypothesis to understand a certain molecular process underlying this disease, the first thing you do should not be, what animal can I use? It should be, how do I understand this disease in humans? What approaches can I use ethically and in an innovative manner to understand the biology underlying the human disease? Why aren't we answering as many questions as possible, as many hypotheses as possible, using non-animal methods? You would expect more from science because it's evidence-based, but I think that scientists are also human, and this is part of our culture as a species. I think we keep doing the same things that keep us comfortable and safe.
And that way of thinking, that culture, it's ingrained in us, like you said. I think throughout history, though, that leads to problems that eventually have to be solved, right? The way we've always done it. Doing something just because that's the way it's always been done, that's not a way to go forward doing anything. It's not even, it's as you suggested, that's not science. That's not evidence-based practice. Evidence-based practice and science is constantly evolving, no matter what it is, not just animals and medicine. It's everything. It's as new information becomes available, you should be asking, if it confirms what you have before, okay, maybe we're on a good model. But if it doesn't confirm it, you need to ask which one of these is wrong, and maybe do a third experiment, look into it, and sort it out. And science evolves, and we need to be challenging it and moving forward. I'm just the method of the idea that we've always done it like this, therefore going forward. I know that's the culture. That's how you make mistakes. That's how you kill people. That's how you hurt people in medicine. I mean, if you went into an operating room and didn't even consider taking someone's gallbladder out with a laparoscopic approach, that's negligent. If you don't know how to do a laparoscopic cholecystectomy, take someone's gallbladder out. At the beginning of the surgical time, that wasn't considered because the technology wasn't there, but it evolves. And if you don't evolve as science does and as change comes, you get left behind. And yeah.
I think accountability in science is not at the level that it should be. And it's really difficult to challenge, right? So when you speak and you challenge the status quo, you're vilified. And I was doing this heart failure work, and I was looking at these receptors that when they interact on the membrane of a heart cell, that they were supposed to, if they were activated in time, that they would protect against a heart attack. So we're doing a lot of different studies, and long story short, while I was doing all of this work, about 4 years into this work, my dad actually had a heart attack. And he required quadruple bypass surgery. So I was with him for, I don't know, well over 3 weeks. He underwent the surgery. And I remember the very next day, or when he was recovering, I sat next to him. I sat next to him for many days. And I remember it was just shortly after his surgery, I was watching him by his bedside. And I was in this ward with many other people. This was at the Halifax Heart Centre in Eastern Canada, in Nova Scotia. And I was looking at him and all the other patients who were there, who had undergone one type of surgery or another. It was a cardiac unit. I think I was forced to answer a question that I had been avoiding for a long time. And that question was, is the work that I'm doing in mice ever going to help people like my father and all these other people in this world? And the answer that came to me was no. A very loud, resounding no. The work that I was doing was never going to help him or anyone like him. And it was a profound realization. It didn't just happen because I was sitting by my dad's bedside and I was emotional or anything. It was also based on my firsthand experience in the laboratory, looking at the scientific literature of the translational failure rate and all the reproducibility issues that we had from lab to lab. And there were so many different issues with it. But then at the end of the day, it came down to, at the end of the day, I did not go into science to cure mice of heart failure, that it had to help humans. And I noticed that this is a real life scenario now. Is that research ever going to translate to humans? It wasn't. And so a few weeks later, I believe it was like 3 weeks later, I was back in the laboratory and I ran into this really a senior researcher who had been funded by the National Institutes of Health to study heart failure in mice. And he'd had a career for like 25 plus years. And so I just saw him and I said, "Hi, Dr. So-and-so. Do you think these receptors were activated in my dad's heart during his heart attack?" And I will never forget the answer that he gave me. He looked at me and he goes, "How the hell would I know? We've never looked at this in the human heart." That sentence changed my career path. That was one of 2 epiphanies that I had where I'm like, I'm done with animal research. Because if you don't even know how that's going to, or that if you in a 25-year career in biomedical research that you've never bothered to look at this in a human tissue or human something, then what are we doing? To me, as a younger scientist at that time, that was the question. And I challenged him on that. Within another year, I think, I left that lab. This is 2026. Do you have a cure for diabetes? I mean, we discovered over 100 years ago, 105 years ago now. So these are questions that we have to ask now. The entire scientific community, I'm not blaming anyone. I'm just saying that why aren't we coming together as a society, as a scientific community to say, and to really accept that animal models are not the be-all end-all, and treat them as such. They are not the only tool in the toolbox, but most of the time I feel like it's being treated as they are the only tool. They're just one tool, a tool that we use too often. What is that expression? If you use a hammer, everything is a nail. That kind of
It's applicable.
That's what we are seeing. So yeah, I mean, I walked away from all that, and I entered into this world of non-animal methods or new approach methods or NAMs. I worked for a U.S. nonprofit called the Physicians Committee for Responsible Medicine. I was their director of laboratory science for a few years. During that time, I had the opportunity to interact with a lot of different stakeholders. So interacting with regulators like US FDA, EPA, European groups, and Health Canada here, to interact with these scientists in conferences and other places. And during this time, I just realized that there's an industry people as well, and academics who were developing these new technologies. And it was just a different community that I had never, ever been exposed to before.
Thanks for sharing that very personal story, Charu. That, like you said, that was kind of a tipping point for you. And it's very hard to question maybe what I've been doing is not serving the need I think it is. And it really gets to the core of your identity. I know I've experienced it, and many others in this space have had some defining moment. And a lot of times it is personal. So I really appreciate you sharing that personal story, and that moved you, and it takes something, and that was what it was for you, and you're on this new pathway now, really trying to make a difference for human health, motivated by your own personal events in your life. And you touched on something in passing that I think a lot of people don't also realize. We talked about the very poor translatability from animal models to human models. You have a result in animals. The probability you're going to have that same result in humans is very low. There's a reproducibility crisis. Another problem, sounds like it might be similar, but it's really not, Would you like to share the reproducibility crisis in animals? I think that's something that I know as an MD, I was not aware of this until recently. I figured if you studied, a group of 100, 100 mice and you introduced a certain drug and you saw a certain effect, I felt like you should be able to do that 10 more times in 10 different labs and get the same result. But that's not really the case, is it?
No, that's not. That's exactly what it is, the reproducibility crisis, that you're not able to take data from one laboratory and create it again in another laboratory. This is a really good example that I could give you about this. There was a human study that identified a certain protein in the pancreas that is thought to play a role in diabetes. So the first thing that, everybody did in this community was they went and made these knockout mice. So they took that gene, they deleted it from these different mouse strains, and they were like, okay, does it increase diabetes or decrease diabetes? Let's see what happens. And they did all these things and multiple laboratories. And these are some of these people had long careers and well respected in their fields. They created these knockout animals and they published this data. And within like a span of 5 years, you saw multiple papers in very high profile journals. And at the end of the day, nothing was reproducible. So some laboratories would say knocking out this particular gene increased glucose levels. So it caused diabetes. The other ones decreased. And not just looking at the blood glucose levels, but looking at many other parameters of glucose regulation, they were up and down and everywhere. Nothing was consistent. But at the end of the day, they all concluded that the deletion of that gene caused increased diabetes. There was an original colony of mice that were in France somewhere, and they had done the gene deletion, but they took these mice and they sent them to 2 laboratories, one in London and one in Toronto. The ones in London did it like 3 times, and the ones in Toronto did it 2 times, and the results were completely different. And so the reproducibility crisis is enormous, and this is just one example. Most of these animals, when they deleted the gene, they saw that these animals got diabetes. And then a couple of years later, they actually did a human study, meaning they found these humans in certain parts of the world that genetically lacked this protein. And so they studied this, and when they studied it, they realized that the lack of that gene in humans protected them against diabetes. So the opposite result of what they saw in the animals. And when those human researchers were trying to publish their data, they were rejected, saying that your human observations do not match the animal data.
It's back to what you said before. That's just ludicrous. That's ludicrous.
So this is, prime example that I often talk about. And I mean, there are tons of it, right? So,
and that speaks to a systemic problem, which we've been talking about to some extent, but the problem is not the researchers. It's not some of the study designs themselves. It's the system that prioritizes animal research as, like you said, the gold standard, despite largely not ever having been validated as the gold standard going back decades for many disease states. We do it because it's been done. And that's what's demanded by federal governments who, at least, the NIH, as I'm sure you're aware, funds the overwhelming majority of biomedical research on the planet, including in the US. And they're still paying for that. They're still funding it. And there's the system is such that largely, like you said, you need to produce animal studies to get something on the path to FDA approval. But it's a system that's deeply flawed that encourages and pays researchers to do these studies and put animals through painful experiments that have almost no chance of producing a safe and effective drug down the road. And might there be some way to change this problem?
So these methods, some of these regulatory testing methods, so whether it's testing a drug or a pesticide or another industrial chemical or cosmetic product, whatever it is, many of these internationally accepted guideline studies, they were never truly validated to see, like back in the 50s and 60s, they were never truly validated to see how well they would translate to humans, because some of the cancer tests they have the reliability of a coin flip. The thing I often say that some of these, chemical testing methods are as reliable as a long-range weather forecast in the Canadian Rockies. And I'm not exaggerating when I say that.
And so just for our audience, we're talking about validity tests being validated. That means, for example, if you expose a dog to a chemical and let's say the dog gets heart failure 100% of the time, if you take that same drug at the proper dose or same chemical and expose it to a human, you're not going to get heart failure 100% of the time. And that's what we're talking about, a lack of translatability. That's a huge part of the problem. So I just wanted to make sure that our audience understands that.
At the time, these tests were not rigorously validated. But now, when you're talking about organ on chip and other methods being validated for human relevance, they are held to unreasonable standards that we can't even meet sometimes. So the animal models are okay at this level, but these new, innovative, more human biology-based, more human-relevant methods, of course, they need to be validated. But they're held to such unrealistic standards that it's delaying the That's not fair. So if you're going to compare two methodologies, then at least hold them to the same standard, that one shouldn't be better than the other. And if it is, or one shouldn't be better, one shouldn't be required to generate data at unrealistic levels. And so why are we doing that, right? And this is what I'm saying. The scientific community, it's puzzling to me. And people ask me these questions all the time. Scientists are really smart. They know what they're doing, which is true. The validation issue is a big problem in the community right now. So all of these things are sort of, I feel that they're all holding us back. And I'm not saying that these new methods don't need to be validated at all, but there needs to be certain guidelines. We as a community should sit down and figure out, okay, if this is an organ on a chip for liver toxicity, what criteria do we need to meet for it to be acceptable to predict human liver toxicity? So those things are happening a lot around the world. Very, very exciting. But this is one of the biggest, frustrating issues that industry and other groups are experiencing at the moment. How do you validate these things? And we are being asked to compare the data coming from these new technologies to animal data. And so if they don't correlate, then it's like your method isn't good enough. But there's a fundamental issue with that, because most of the thinking that goes into developing these new technologies and using them to emulate human biology, these methods are not based on the same thinking process as when you take an animal, right? You take an animal, feed some pesticide, let it die, take out, open up the body, look at the organs to see what happened. But with these new methods, it's a different kind of thinking. So you can't directly compare the data from some of these new technologies to animals, but we are still being forced to. Still need animals. My biggest issue right now, I'll be honest, and maybe this is one of the other take-home messages, is that animal models are not the be-all end-all. When Descartes thought that animals were machines, this is a time when we know these are sentient beings. And so if you're going to induce a 3-hour heart attack in dogs, and this is real, this happened in Ontario a few months ago, there was an expose. There were some whistleblowers that came out from a heart attack laboratory where they were taking these dogs and giving them 3-hour heart attacks, complete block heart attacks. So they would tie off their artery for 3 hours and they go into these massive heart attacks. And then they use these animals to study different aspects. They were doing some imaging, like MRI imaging, to see how the cellular processes change in the heart, how the heart recovers from heart failure, etc. What is the cost of that? I think that at this point in time, we as a community need to come together and evaluate the science and the ethics of it all.
Very complex, for sure, Charu. So much has changed in medicine since the 1930s and 40s, when animal testing became mandatory for getting a drug FDA approved. Should we really be funding those studies still and relying on some of the same methodologies and the same poorly translatable studies, poorly reproducible studies that have severe ethical questions and challenges with very little guardrails around them for parameters for performing the studies? Should we really be relying on that old way of doing things just because we've always done it? Or should we be evolving? Should our taxpayer dollars be funding research which is human centric, which has some ethical guardrails around it? Should we be asking at what cost? Is it we need to get a better drug on the market to mitigate symptoms of Alzheimer's disease at any cost? So these are some important questions I think to be asking. And if you're like me and perhaps like Charu, you're coming to realize that animal testing for biomedical research and discovering treatments and toxicities in humans, it was the past. It's not the future. We have better ways to do it, more cost-effective ways, more human-centric ways. We touched briefly on NAMs or non-animal methods or new approach methodologies. They're evolving quickly, and until a lot more money is invested into them, it's going to be hard to get to where we want to be with being able to validate them in a timely and efficient manner. As a reminder, we're talking about animal testing, but before getting approved by the FDA or the equivalent agency in Canada or whatever country, any drug still has to be tested in humans. So that's going to happen 100% of the time. And so it's not just animals up approved in a drug, let's approve it. It still goes through human trials. So human testing happens. Question is, do you want to go through animals as the preclinical testing, which, as we've touched on and I've talked about in prior episodes, has a very poor translatability rate. The results that you find in animals do often not translate to humans. You get unexpected side effects, you get failure rates, which are very high, and that's why we're well north of 90% in terms of clinical failure rates for drugs that prove safe and effective in animals. We have better models. We have a severely flawed system in place that still rewards this kind of work, which has a very low probability of achieving something meaningful to get into the doctor's hands to treat their patients. We have better options, and a shift needs to be made. If you felt like this touched you, there's plenty of information out there that you can find, and I encourage you to speak with your lawmakers and let them know how you feel. If you feel like there needs to be some kind of guardrails in place, if we're going to continue with testing on especially dogs and cats, the animals we have in our homes, or any animals, and if you let them know, and if you feel like you agree with me that this is the past, not the future, you want your taxpayer dollars invested in human-centric research methods, not animal methods of 80 years ago, let them know. Let them hear it so that organizations like Charu's and so many others, which are working to move science forward to help humans more quickly, let them know. Charu, I wanted to give you the floor here. Last few minutes, anything you'd like to close with, final messages to impart?
Now, I wanted to say that we all have a role to play. This is not just a science issue for scientists to deal with, right? It is a societal issue. When you have that kind of a failure rate, when you're waiting for a cure for a loved one, this is, you have that responsibility. And like you said, it is tax dollars. So I think what it's going to take to change this culture of science from mouse models being the gold standards for human biology to human-based models being the gold standard for human biology, it's going to take a lot. It'll be a bottom-up and a top-down approach. So bottom-up meaning, scientists who are starting their laboratories or developing another next grant proposal that they themselves are like, I'm going to adopt organ-on-chip technology or some other thing. So there's, these types of change, or a certain university having more funding for a particular new method. And then there's the top-down approach, people at the top, so the lawmakers and funding agencies and and publishing companies and all of them saying, we have certain restrictions on animal models, that you must show some human relevance, that you must include human data and show that human biological relevance. But I think at this stage, and this is the example that I didn't really get a chance to talk about, but I wouldn't say much about it other than to say that people should always reach out to their lawmakers and policymakers. And this happened in Ontario with those heart attack dogs. And we saw footage of these animals that were released by, and then there was public outrage. And the Premier of Ontario, equivalent to a governor in a US state, he actually said, I'm going to ban dog and cat testing in Ontario. So as of May 14, 2026, he was able to amend our Animals for Research Act and ban the use of invasive research on dogs and cats, with one narrow exemption, that it is allowed for certain veterinary applications to do clinical trials for medicine and other things to benefit cats and dogs, and for veterinary education. But other than that, you can't use dogs and cats for whatever you want. And to feed them pesticides and give them 3-hour heart attacks, that is no longer allowed in Ontario. And so I think that we have a role to play. Everyone who's listening to this, if you really want to do something, write to your lawmakers, write to the policymakers, write to funding agencies, write to scientists. And don't be afraid to question the status quo. Every single study that has happened, and this is one of the things that I really had to deal with, everyone's like, we found insulin because of dogs, and it was one of Canada's greatest contributions to humanity. Yes, that was in 1921 when we didn't have better methods available. If you're using an iPhone 17 today and you refuse to use a rotary phone, then don't tell me that I have to use dogs today. And if you ask some of my students that I've trained, they will tell you how to discover insulin without using a dog today in 2020.
Not the only way, right?
Yeah, so I think it's really important that all of us as a society come together and work together. This is how we've advanced everything. And there's no reason why we can't quickly move away. Otherwise, at this pace, I mean, it's going to take a long time to get there, but there is a way to get there faster.
It doesn't have to take a long time. It doesn't have to by any stretch.
Yeah.
So a couple of points we'll close. Question the status quo. That's how progress happens. Not just in medicine, across the board. Question the status quo. Is the way that we're doing it now still the best way? Was it ever the best way? Or are we doing something now just because that's the way it's always been done? And new information becomes available, you adapt or you die. That growth happens when you take in new information, you evaluate it, and you question what you've been doing. That applies here. And as the example that Charu laid out, a huge success in Ontario, a combination of multiple factors, but it was the people, right? The people speaking up in response to documentation of what's really going on, transparency. The people spoke and said, this is unacceptable, and the government acted upon it. So speak up, make your voice heard, work with your local nonprofits, work with your governments to let them know how you feel and help make change, move forward. So that's how progress is made, which in what we're talking about, it's really for the benefit of human health. Charu, thank you so much for joining. This was an animated, lively discussion. I learned so much from this, and I'm pretty sure our listeners learned something too. Thank you so much for being on here, Charu.
Thank you so much for having me.
Thanks for listening. These conversations aren't always easy, but they matter, and they're worth having. If you enjoyed this episode, you can find more information, videos, and updates at johnliebermanmd.com. Take what resonates, ask questions, and keep learning. The content of this podcast is for informational and educational purposes only. It is not medical or legal advice and does not replace speaking with a qualified professional. Always seek guidance specific to your situation.